Hello, Professor. I'm learning about viruses and I just wanted to ask, what makes coronavirus so different from other viruses?
Well, a lot of aspects of this virus vary from other viruses, such as virion structure, genome structure, virology, and more.
Okay, but why are they called coronavirus?
Well, that's a very interesting question. They are called coronavirus due to their prominent feature, which is the club-shaped spike projecting from the surface of the virion, like a solar corona. Coronavirus virions, or viral particles, are spherical and contain spikes, membranes, envelope, and nucleocapsid proteins. However, what else could determine the divergent behavior of coronaviruses?
Well, I know that viruses contain a genome. Could that by chance be another aspect that determines its divergent behavior?
Very good. For instance, let's talk about SARS-CoV-2. It has a large genome which is 5' capped and 3' polyadenylated. There are two flanking untranslated regions, or UTRs, and it contains several open reading frames that encode multiple proteins. Therefore, at the beginning of each structural and accessory gene, there are transcription regulatory sequences that are required for the expression of each gene. Also, there are enzymes responsible for replication fidelity that proofreads RNA during replication, for example, NSP14.
Wow, that is really fascinating. Besides that, I understand that viruses are in need of a host cell in order to utilize the cell's machinery for replication and proliferation. However, I've heard that COVID-19 spreads really easily. Can you explain how they infect?
Well, where should I start?
Let's say initial attachment. How does this occur?
The S protein interacts with its receptor to attach the virion to the host cell. The access to the host cell cytosol is due to acid-dependent proteolytic cleavage of two sites within the S2 portion to permit fusion. In the case of SARS-CoV-2, the S protein also has a polybasic cleavage site, PRRAR, at the S1-S2 boundary, which permits efficient cleavage by the prototype proprotein convertase furin.
What happens after the viral attachment?
The open reading frames 1A and 1B RNAs are made by genomic RNA and then translated into polyprotein 1A and 1AB respectively. Polyprotein 1A and 1AB are cleaved by protease to make a total of 16 non-structural proteins.